If you are a returning subscriber, welcome back. If you are new, welcome to my channel. My name is Dr. Shingu Mupfawa. Guys, if you haven't subscribed, please do so. We want to create a community here. In today's video, we are going to talk about the DNA replication process step by step. If you are preparing for a life sciences or biology test, this is the perfect place to start. So, what is DNA replication? DNA replication is the process by which the DNA molecule makes an exact copy of itself. It occurs during interphase of the cell cycle before the cell divides, and it happens inside the nucleus of the cell. Now, let's break down the process of DNA replication step by step. Firstly, it starts with the double helix unwinding and unzipping. Then the weak hydrogen bonds between the base pairs break to form two separate strands, and each strand acts as a template for the formation of complementary DNA strands using free-floating DNA nucleotides found in the nucleoplasm, where adenine pairs with thymine and guanine pairs with cytosine. Two identical DNA molecules are formed. Guys, please note that though this process is controlled by enzymes, the examiner does not require you to know the names of the enzymes. And also, when explaining the process, don't forget to write DNA double helix for the first point. Remember that the two strands or both strands are used as templates. When discussing the use of free-floating nucleotides, don't forget to mention that they are free-floating DNA nucleotides. Don't just say nucleotides. Now, let's look at the significance or importance of the DNA replication process. Replication is important because it ensures that the genetic material is doubled and that the two identical daughter cells that are formed after mitosis are identical to the parent cell. That's your quick guide to DNA replication. If you found this video helpful, drop a comment, like, share, and subscribe. See you all in the next video. Bye.